Alright, the last video for this series, we're going to talk about the oxidative phosphorylation and the electron transport chain. So I'll write those off the top. Electron transport chain plus oxidative phosphorylation. Right, that's some really big words, but we're going to break it down and make it nice and simple. Let's start off with where this is happening, just to give you a sense of perspective. We're inside the mitochondria. Okay, and the mitochondria has these lovely folds inside, so let's draw them in. And let me grab another colour. Okay, so we have got some bits to label. This is the outer membrane, and this is the inner membrane. This inside of here is called the matrix. It does make me think of Neo with dark glasses, so that's showing my age. The matrix. And then inside here is the intermembrane space. Okay, so we were just talking in my previous videos about the citric acid cycle, which takes place inside of the matrix. So this is where the Krebs cycle or the citric acid cycle, C-I-C-L-E, where the Krebs cycle is happening, it's in here. Now we're going to talk about the electron transport chain and oxidative phosphorylation. Those are happening between the inner membrane, the intermembrane space, and the matrix. So we're going back and forth across this membrane to make it all happen. So this is where the ETC, we'll call it, electron transport chain, and OP, oxidative phosphorylation, is happening. Now let's draw a picture of what this membrane looks like. Now you may know that we've got a phospholipid bilayer. So we've got these phospholipids with their non-polar tails sticking into the middle, which is creating this inner membrane. We've got some cool proteins that go across this bilayer, and I'm just going to draw them in as I'm going. Okay, so we've got, we'll call this guy number 1. Then we've got number 2, which doesn't go all the way to the top and has a little hat on that we're going to call Q, like the Queen's crown. So I've got some more of these tails sticking in. Great. Okay, now we're going to keep going, and we've got number 3, which does go all the way across. And put our phospholipids back in there. Great. Number 3, and then we've got a funny wee hat up here. I can't remember what that's called, so you have to look it up. And we've got a tooth-shaped one over here, number 4. Put our little tails in. And then, very importantly, we have this big box here, which is called ATP synthase. So I'll put that name in, and this is the coolest enzyme ever. I absolutely adore it. It has got, its first piece here, it has got like a axle on a motor, and then it's got another piece down here. And it's actually got this other piece that kind of joins these two together, like that. So we'll call this ATP synthase. Cool. Now let's find out what happens in this space as we have the electron transport chain active and oxidative phosphorylation creating ATP. So let's start with our wee friends from before that were created during the Krebs cycle and glycolysis and various stuff. We've got our over here, NADHs, and we've got our FADH2s. And these guys are coming in to drop off their electrons and their protons. So let's start with NADH. So it comes in, hits protein number 1, drops off some electrons and protons, and leaves as a lovely little NAD+, ready to support the rest of respiration all over again. What happens to its electrons? Let's have green for electrons. So its electrons go do, and each stage they lower in their potential energy as they go through. So they go from 1 to 2, up to Q, over here to 3, across this top hat thing that I've forgotten the name of, down through 4, and out as some E minuses. They're ready to attach to some oxygens to make some water. This is why this process is aerobic, because we need to have oxygen available in order to make water, in order to drive the entire chain. Okay, but we're not ready to make water yet because we're missing hydrogen. I'm just going to finish off the other electrons. So we have FADH2, which jumps into number 2. It can't go into number 1, but it can go into number 2. So it goes in there, comes out as FAD, having dropped off its hydrogens and its electrons. So they go up through number 2 and follow this journey. Actually, I think I made a mistake before. I think number 1 goes straight to Q and then number 2 goes up to Q. So let's get rid of that one. Okay, anyway, they go on this journey and they contribute to these electrons that have come out. So that's the electron transport chain. But what does that power? What's the point? So the point is that it's powering an electrochemical gradient to be created, which will power ATP synthase to create ATP for us. So let's talk about these protons that got dropped off. So the H pluses are protons, and they're getting pumped out of all of these lovely proteins by the power of each of these electrons as they drop in energy going from protein to protein. So there's a few of these hydrogens floating around here, but most of them are over here. Now I've forgotten to label these, so let's put some labels in. We've got in here the matrix, which we saw up here. Out here we've got the intermembrane space. Great, so we're going between the matrix and the intermembrane space like we talked about before. So we're pushing these hydrogens out into the intermembrane space to try and create a gradient. So if we've got more H's over here, here's our protons, then there's a natural gradient between chemicals, so the hydrogens and the hydrogens, but also between the charge. It's way more positively charged over here and not as positively charged over here. So for two reasons, both the positive to negative and the hydrogen is more and hydrogen is less, these protons, these hydrogens, really want to come back to the matrix site. But these are all one-way tickets out to the intermembrane space. So the only way to come back is through ATP synthase. So let's say this little hydrogen here goes in the entrance of ATP synthase. It gets pushed around and comes out the exit of ATP synthase. This is the only way it can get back into the matrix. So all of these are all rushing over here to try and go through this ATP synthase, causing this rotor thing to turn, which is amazing. And that changes the conformation of this side of ATP synthase that literally squishes together ADP and a little phosphate to give us, ATP. Yay, ATP. So ADP and phosphate go in, they get squished together by the turning of this amazing rotor. Definitely recommend you look that up on YouTube because it's cool. And then ATP comes out. And as this goes, it's just churning out ATP. Meanwhile, the lovely H's, H plus, got two of them. So half an O. All right, so we've got two electrons, two H pluses that have come through ATP synthase. We've got plenty of oxygen around because we've been breathing, and that makes water. And because of the oxygen, because this is all going, it allows everything earlier in the chain to continue. If we didn't have oxygen, we'd have to rewind all the way back to glycolysis and go off down one of the anaerobic fermentation routes instead of getting to go through the Krebs cycle and end up with lots and lots of ATP from this super cool enzyme. So I hope that helps.